wonderful October gallery for another one of the Ecology, Cosmos and Consciousness lecture series. We meet here every last Tuesday of the month, more or less, and it's been going on for about 3 years now. Some new faces, some old. I'll just make a few quick announcements. We've had a series of talks on alchemy. Tonight we're going to be talking more about chemistry and neurochemistry. But next month there'll be another talk on, actually a performance, in fact, on alchemy by the extraordinary Oriel Defenestrate Bascule from Australia, who's giving a performance on Solve e Coagula. That's an alchemical mystery play. And then we'll be back to another talk about psychedelics again. After that, I'll be giving a talk about Huichol Indians in Mexico and their use of peyote. So do come along to further events. We have a Facebook page, you can see what's happening on there. Oops. Tonight's speaker, Dr. Robin Carhart-Harris, did his PhD in neuropsychology, I believe, or something.
Psychopharmacology.
Psychopharmacology at Bristol University, under the supervision of our ex-Ruxar Professor David Nutt, and then moved with David Nutt to Imperial College. And for the last few years they have a research project there, the Beckley Imperial Psychedelic Research Project, where for the first time really, there's been a little bit of research been looking at brain imaging and psychedelics, primarily in Zurich with Vollenweider, but there hasn't really been much done since the 1960s. And of course the technology has advanced massively since then, so we're very lucky to get to hear a little bit of how psychedelics work in the brain. This has been kind of virgin territory really at this current time. So without further ado, please help me in giving Robin a very warm welcome.
Thanks very much, Dave. Thanks everyone for coming to listen. So it's going to be quite a relatively dry science talk, so hopefully not too much of it will go over your heads, but I'll try and do my best to make it as interesting as possible. There'll be lots of pictures of brains at least. So first of all, to start off with, give you a sense of the research that we've been doing over the last couple of years. Really, most of our work and the work that's progressed the most and that we've done the most analysis on, now published on, is our fMRI work with psilocybin. So if you don't know what psilocybin is, it's the active component of magic mushrooms. We've also got an ongoing MDMA fMRI study. So again, if you don't know what MDMA is, it's ecstasy. It's better known as ecstasy, although some would contest that link. But, for simplicity, we'll say that MDMA is ecstasy. And we've also recently completed a psilocybin MEG study. And we've got all the analysis packaged up, all the data rather packaged up. And so we've just got the rather large job of analysing all of that, but that's quite exciting. I'm sure we're going to find something especially interesting in that. So the basis of the talk is going to be the psilocybin fMRI work that we've been doing and the analyses we run on that. And I'll end the talk with some very hot off the press analyses we've done with our ongoing MDMA fMRI study. So this analysis was actually done yesterday, and the images made today, so they really are genuinely fresh results. Okay, so to proceed. So before we threw anyone in the scanner and gave them some potent psychedelic drugs, we wanted to tread carefully and make sure that we were giving a dose that was tolerable and that people were going to manage this, because not only is the other drug effects profound, but also the environment that we're throwing them into is quite potentially anxiogenic. It's quite a strange, unusual environment. If you suffer from claustrophobia, it can be a bit challenging to go into this restricted environment of the MRI scanner. They're also very noisy, and this can be perturbing for anyone even in sobriety. So we simulated an MR setting. It was actually much more crude than this, but this gives you an idea of the dimensions of the MR scanner. We gave our plan was to give intravenous psilocybin. So really the rationale for that was, there were a number of good reasons, but it was very suitable for imaging because the onset of the subjective effects is very quick, it's very rapid, and fMRI particularly is quite a sort of sluggish modality. So if you want to map changes in brain activity, it's very difficult to do that with fMRI if your subjective effects are coming on gradually. So with intravenous administration, we could really get in there and very suddenly alter consciousness in a profound way and then map these effects. Also, in terms of management, the effects last for a shorter duration, so that's easier to manage rather than have someone in your lab for a few hours hallucinating and experiencing strange subjective effects. So we dosed 9 healthy volunteers, 2 of them were females. We started off at 1.5 milligrams of psilocybin. We gave it as a 60-second bolus injection in 10 mils of saline. But these effects were quite mild, and we sought permission after having in our protocol that we'd give up to 1.5 milligrams. We sought permission to give up to 2 milligrams, and subsequently that's the dose we've stuck at. So that produces quite reliable, quite strong subjective effects, but they're very well tolerated in all our subjects so far. And actually we dosed, I think we've done over 50 sessions now with intravenous psilocybin, so we know quite well that it is well tolerated. Okay, so to give you a better picture of the dynamics of the subjective effects, the pharmacodynamics, in grey you can see the time course of the effects with 1.5 milligrams of psilocybin. I do have a pointer here. And so in black we have the time course of the subjective effects with 2 milligrams, and what's important to see is here's the infusion, 60-second infusion. By the end of the infusion at 2 milligrams, they're already starting to feel something. It's incredibly rapid onset of effects. Do you look familiar? By the following minute, people are up to near peak, so it really is a very rapid onset of effects. People describe it as a rush, which is quite unusual to have a psychedelic described as being like a rush, but yeah, that's what they say. And so the duration, you get good effects for 20 minutes, at least half an hour. So you can really get quite a lot of good imaging work done with this mode of administration, with this dose. So this is just to give you an idea of the relative intensity of the effects, because some people doubted really that with intravenous administration in this dose that you could get some of the profound effects that you do with oral administration. So one of our volunteers said that was undoubtedly the most intense psychedelic experience he'd ever experienced, and so he really did emphasise that it certainly wasn't a walk in the park, and this was really quite a profound experience for him. So to give you a better idea of the range of the subjective effects, we gave our volunteers a number of different items. So here are the top 10 highest rated items. We actually gave them around 30, but this gives you a good idea of the typical effects really. So the first one there, I saw my surroundings change in unusual ways. In blue you see the ratings under psilocybin, in grey it's placebo. And the top anchor is much more than usual, bottom anchor is no more than usually. And to give people a sense of what that meant, we said that the bottom anchor, no more than usually, refers to normal waking consciousness. So anything above that is an altered state. So I saw my surroundings change in unusual ways. Incidentally, you can see the placebo, they are up a bit. So when you administer placebo, people are often looking for some effects. So they're not always entirely convinced that they haven't had anything. They're much more convinced when you give them the drug. It's hard to doubt that you've had something when you do give them the active compound. So the first one, I saw my surroundings change in unusual ways. I saw geometric patterns, that's very classic of classic psychedelic drugs. I felt unusual bodily sensations, tingling, this kind of thing, often described. Things look strange, very simple, but that's often what people say. My imagination was extremely vivid. People often talk about actually their imagination colouring what they're actually perceiving, so there's a kind of overlap, a kind of merging of modalities, which is highly unusual. My sense of size or space was altered. People sometimes refer to this kind of Alice in Wonderland effect. They might feel like they've gone small, or they might equally experience a kind of expansion. They might describe the scan of aura as becoming expanding and seeming wider. Sounds influenced things I saw, this kind of synesthesia type experience. My thoughts wandered freely. That's very typical again. People talk about this kind of loosening of their thoughts and of their imagination, and of all modalities really. Perception becomes loose as well. My sense of time was altered. That's very typical and very hard to fathom if you don't experience this profound altered state of consciousness. The experience had a dreamlike quality. That's another thing that comes up quite reliably with psychedelics. And it's quite interesting, and there's some biological evidence to suggest that the neurophysiology of rapid eye movement sleep or dream sleep shares some similarities with the psychedelic state. That's kind of interesting that there may be a literal biological overlap between these two states. That's actually something we'd like to explore. So, to move on to the actual imaging study and the design, the first study we did, we actually ran 2 fMRI studies with psilocybin. The first study we used arterial spin labelling. This is a measure of brain perfusion or blood flow, and which is a correlate of brain activity. So if there are increases in blood flow, on a simple level we can infer increases in brain activity. There were 15 healthy volunteers, all had previous experience with hallucinogens. That was a safety consideration. Mean age of 34. So the scans were 18 minutes long. There was 2 of them. With the ASL study, we did the placebo scans first, followed by the psilocybin scans. So it was a fixed order in this case. Eyes open, task-free rest. So rest is slightly, said in inverted commas because people are lying there relaxing. It's a task-free condition, but we're never truly at rest. The mind will be wandering and will be daydreaming about this and that. And we gave 2 milligrams psilocybin, so you saw the dynamics of the subjective effects with 2 milligrams of psilocybin. Okay, so to give you a quicker overview of the design, essentially we're looking at changes in perfusion post-infusion of the drug or placebo. So we make a contrast, for simplicity I'll say a contrast with post-injection, post-infusion of psilocybin and blood flow in that time period versus the blood flow pre-infusion before we gave either the drug or placebo. And then we can take that subtraction from both conditions, so subtract post-psilocybin from pre-psilocybin and post-placebo, which is actually a saline infusion, versus pre-infusion of saline. So we're subtracting a subtraction essentially. And this is where we found changes. So there had been a little bit of imaging work done already with psilocybin. This was an FDG study that measures changes in glucose metabolism. They'd actually found increases in some regions of the brain. So on that basis, we were expecting really some increases, but this isn't what we found. We found decreases, and we only found decreases. Even dropping the statistical threshold, we didn't find anywhere in the brain where there were any increases in brain activity. This was slightly surprising. We actually sort of double, triple, checked a number of times to make sure that we didn't get the signs wrong and this kind of thing. And subsequently, we have repeated the study, albeit with a different fMRI modality, and we have found the same results again. So we feel relatively confident now that this is what's happening. This is what's accounting for the subjective effects of the drug. We've also controlled for changes in physiology, heart rate and breathing, to see if these effects were accounting for the brain effects, and they're not. So we've controlled for that, and we feel relatively confident now that this is what's happening in the brain to account for the subjective effects of psilocybin. So what's also interesting is that the decreases were localized to particular regions in the brain. In the middle, I wonder if I can get the pointer up. is the thalamus, and there were bilateral decreases there. The posterior cingulate cortex, which I'll talk about in a moment, is an important cortical hub structure. Medial prefrontal cortex and along with the cingulate cortex. Also bilaterally in the putamen here. I'll say a bit more about these regions to give you a sense of what decreases in these regions mean. This always adds another compelling dimension if you have correlations with the brain changes and subjective effects. So here we see that in the anterior cingulate cortex, the larger the decreases in blood flow, the more intense were the subjective effects. So that was a significant effect. When we looked in the thalamus, it was trend level, so again, a trend suggesting that the bigger the decrease in blood flow in the thalamus, the more intense the subjective effects. And the same was true for the posterior cingulate cortex. So to give you a feel of the subjective effects with some first-person accounts, albeit very brief. This volunteer said there was a definite sense of lubrication, of freedom, of the cogs being loosened and firing off in all sorts of unexpected directions. So that's quite a nice description because it does fit in with the brain effects that we saw, and I'll describe that in a moment. Also, time became another meaningless concept, so appealing to this disturbance of time perception, which is so common in psychedelic experiences. I could have been there a minute or a million years, he said. So that was real ego death stuff, a total dissolving of the ego boundaries. I only existed as a concept or as an idea. But again, it's very hard to fathom it, and we carry so many assumptions. We have assumptions about ourselves being agents in the world, about having a coherent personality and such like. And it's one of the most profound things that people describe with psychedelics is that they observe that thing that they have so many strong assumptions about break down in the psychedelic state, and that can be really quite profound, and it can account for things like these spiritual like experiences where there's a loss of the self, a breakdown of the ego. And finally, it was certainly quite difficult at times to know where I ended and where I melted into everything around me. So this is appealing to what's sometimes described as ego boundaries, or having a sense that you're an agent being kind of apart or separate from the world. And again, under psychedelics, sometimes people describe a sort of dissolution of this subject-object differentiation. And again, this can account for some of the spiritual type experiences that people report with psychedelics. So as I said, we repeated the design to some extent with another fMRI modality, this time bold fMRI, which measures brain activity essentially. 15 healthy volunteers again, so same sample size, same dose of the drug. Placebo-controlled design. There were 2 scans on this occasion, 7 days apart, and we controlled for order, in case order could have been accounting for the effects that we saw. So by that I mean that half of the volunteers had psilocybin in their first scan, and half had placebo. So the order in which they had psilocybin was balanced. In this case it was eyes closed rest, so they simply lay there with their eyes closed and relaxed. And for simplicity, the contrast here was the changes in brain activity, we'll say, post-infusion or injection of the drug versus pre. And so we did that for injection of psilocybin and injection of placebo, or saline. And the changes we saw, there were again decreases. Again, we didn't see any increases in any brain region. So here we can see there's the posterior cingulate cortex again, the ventral posterior cingulate cortex, medial prefrontal cortex, some putamen effects and insular, and along the ventral visual stream, or what's sometimes referred to as the what pathway, or what things are in our environment. So closely related to perception, visual perception. And here I've registered the decreases in brain blood flow as we saw with ASL, so the results I showed earlier, and the decreases in BOLD. So these are the consistent decreases that we saw across the 2 studies. And you can see that there were consistent decreases in the ventral posterior cingulate, in the medial prefrontal cortex, and bilaterally in the ventral thalamus and the putamen. Okay, so what of these regions? Well, as I've said before, they're important hub structures in the brain, and we can map this, or we can tie this up with a phenomenology where one of our volunteers said there was a definite sense of lubrication, of freedom, of the cogs being loosened, and firing off in all sorts of unexpected directions. So the explanation I'm giving here is that the function of these regions is to constrain cognition or keep it within certain bounds, keep a certain regularity and familiarity to our cognition. And if that breaks down, then we would experience the world as strange. There would be an unconstrained quality to our perception and our cognition. So that's the explanation that we're offering for the decreases that we saw in their location. So psychedelics take out the brain's hub. So what more about that? Well, just to give you a better feel for the role of these regions as hub structures in the brain. Let's keep it simple, even though it's not simple. So the posterior cingulate cortex here is this region that you can see on the left column. And here it's been fractionated by a certain analysis technique to find different bits of it that might do different things, essentially. And then if you see the networks which are on the third column along, and there's all these different networks. Well, these are called resting state networks or intrinsic networks in the brain. They all subserve slightly different functions in the brain, actually sometimes quite dramatically different. You know, you have a visual network, you have an auditory network, a motor network. You have ones for more high-level things like our sense of self, potentially, introspection. And then you have other ones that are quite generic, kind of sometimes described as task positive or attention networks, which engage reliably when we do anything that requires our attention. I'll come on to that a bit more later. What's interesting here is that all these networks are represented within this hub structure, the posterior cingulate cortex. So they're all included, all these different networks across the brain. You can find regions within the posterior cingulate cortex where activity is coherent with these different networks. Also, if you map the structural connectivity in the brain by tracing with another fMRI, or MRI rather, technique, and trace the white matter fibres that connect grey matter, or the neurons in the brain, together, then you will find that the posterior cingulate cortex, the medial prefrontal cortex, and these kind of lateral parietal regions have very dense connectivity, very dense anatomical connectivity, again supporting the idea that there are hub structures in the brain. And just to remind you, those are the hubs that decreased in activity after psilocybin. So the default mode network, you'll hear a lot about this in the next few slides, and it's also a particularly hot topic in neuroscience, I think for good reason, and hopefully I'll make the case to you why it's an important topic that we should take very seriously. So it was first identified because a whole swathe of different tasks that people would ask volunteers to do, in this case PET scanner, reliably deactivated certain regions in the brain. So whatever the task, if it required attention, it would decrease activity in a certain patterns, certain regions of the brain, reliably decreasing activity if you engaged in a attentionally demanding task. So that seemed interesting, and it seemed particularly interesting when it was realised that these regions are the most active regions in the brain. Metabolically, they consume the most energy and receive the most blood flow. So that's kind of curious if these regions are so active during whatever the condition, resting conditions, or even they're also active when we do engage in a task. They just relatively deactivate, but they're very, very active regions in the brain. And so it raised the question, what are these regions doing? You know, they must be accounting for something very important. Their activity decreases when we attend to something, but the actual increases in activity that we see in the brain are only of a very small percentage when we engage in something requiring our attention. Yet these regions which receive so much metabolism and blood flow receive comparatively much more energy than you see if you see a little flicker when you give some kind of cognitive task. So what are they doing? So these kind of considerations have led to people talking about the function of this network and these regions as being like the brain's dark energy, accounting for maybe unconscious processes or very high level processes which make up something, some kind of emergent property at the highest level. Again, another very useful way that we can get a feel for what these regions might do is when we look for certain conditions that might elevate or increase activity in these regions. And I'll describe some of those in a moment. Another important factor here is that these default mode regions, these nodes of the default mode network, have undergone the largest evolutionary expansion. So they're much bigger in humans than in lower primates, or rather higher primates. Default mode network activates during a number of high-level human functions, as I said. And so what are they? Well, predicting the future, or prospection, so that ability that we have to look far into the future and make very abstract but very high-level kind of considerations of how things might play out in the future. And that's something that you could argue is species-specific, that we have that duration, that extended ability to look into the future. Also making social and moral judgments, again something which is arguably species-specific, at least to the extent that we do it. And contemplating our past, it's another thing. Another one is self-referencing, so when we are self-conscious or making self-referential judgments, then you'll also see an increase in activity in these default mode regions. So all these factors and this one especially has led to the idea that this default mode network might be related to the sense of self, or it may even be the network upon which our sense of self rests. It's been described recently as like an orchestrator of the self, or like a conductor. And so, I'm describing the default mode network in such detail because the decreases that we saw in psilocybin were in these regions. And so, if you imagine the conductor of an orchestra being very important for the overall sound, then if the conductor was to leave the room, we would have an altogether more chaotic sound. So that metaphor might help you understand potentially, what's happening in the brain to explain the drug effects. Also, it's been, if you're interested in the Freudian framework, then this network has been linked to the ego. So what else are the default mode network? Well, so regions within it have coupled activity or coherent activity. That's to say that activity in sometimes spatially remote regions like the posterior cingulate cortex, you can see at the back, and in yellow it's written PCC, and the medial prefrontal cortex at the front there, well, their activity is synchronous, it moves up and down, suggesting it's working, these regions are working together as a coherent network. What's also interesting is that activity in regions of a network which activates when we focus, when we engage our attention, these regions and that activity here has a kind of antiphase or competitive relationship with the activity in the default mode network. So there's activity in the default mode network, which is related to kind of introspection, the sense of self, autobiographical recollection, that kind of inner thought, has a kind of competitive relationship with activity in regions which are involved in sort of outward thoughts, or not outward thought, but outward attention, external attention, engaging in the external world. So that kind of reciprocity or competitive relationship between these two networks makes intuitive sense. That we have a network for sort of introspection, one for looking and attending to the outside world, and that they have this competitive relationship, this natural competition. So it really explains why we can never daydream and focus at the same time. It's always either or. So there's a relationship between default mode connectivity and personality. So the higher the connectivity within the default mode network, the higher the neuroticism. This was found in a recent study. Also, the lower the connectivity between a region of the default mode network and proximal regions of the default mode network. So the lower the connectivity in this case, the higher the agreeableness, and also the higher the extroversion. So you'll see a relationship here where the higher the connectivity, the higher the constraint, if you want, within the default mode network, the higher the neurotic traits, and if that network if that connectivity, the strength of that connectivity is less, then the higher these agreeable extroverted traits. So you might predict that there would be a relationship here with depression, and there is. So here looking at connectivity or coupling strength between the posterior cingulate cortex here and this region, the subgenual cingulate cortex, which is heavily implicated in depression. The stronger the connectivity, the stronger the synchrony and the constraint in this network, it's significantly higher in depression, and there's a correlation between scores on rumination. So the higher the rumination, the higher the connectivity within these regions of the default mode network. So this is interesting. You can hopefully see a coherent story emerging here. The higher the connectivity in the default mode network, the higher the neurotic traits or potentially depression, depressive symptoms.
So what do you mean by rumination in that context?
Well, it's a subjective rating scale. So it'll be a number of items which.
But is it a form of introspection? Is that how you mean?
Brooding,
Yeah.
for instance. Yeah. So we'd have to look at the items really to get a proper feel, but it would be kind of brooding and introspection, excessive introspection. It's kind of a coping thing as well. How would you cope with adversity? And would you brood on the adversity, or would you be able to move on? Yeah. So these are effects on global brain function, but very quickly, let's go right back to the beginning and give you a sense for the pharmacology. So here's our Liberty Cap mushroom, where you find psilocybin and psilocin. So psilocybin is actually the prodrug of psilocin, which is the molecule which sticks to the serotonin receptors, and the effects occur downstream of that. So once psilocybin's entered into the body, it seems to be dephosphorylated very quickly into psilocin, and it's psilocin which has affinity for certain serotonin receptors, and the brain effects that we've seen with fMRI seem to occur downstream of that. What's interesting is the similarity between the molecular structure of psilocin and the endogenous neurotransmitter serotonin, 5-HT. So it's quite striking really that a subtle change in molecular structure conferring a subtle change in pharmacology can have dramatic effects on consciousness. So what's the important serotonin receptor that accounts for the subjective effects or the hallucinogenic effects of hallucinogens or psychedelics? Well, it's actually the serotonin 2A receptor. And so it's been found that there's a strong correlation between the affinity or the stickiness of a hallucinogen or a psychedelic for the serotonin 2A receptor and its potency. So to give you an example, LSD has a very high affinity for the serotonin 2A receptor. It's very sticky and it's very potent. So that simple relationship helps us understand to some extent the pharmacology of psychedelics. Where is the serotonin 2A receptor? Well, it's very much a cortical receptor, densely expressed throughout the cortex, but particularly in multimodal, high-level cortical regions like those that are part of the default mode network. And actually, the highest density of the serotonin 2A receptors is found in the posterior cingulate cortex. So on a more reduced level, a lower level, where is the serotonin 2A receptor? Well, it's actually found in layer 5 pyramidal cells in the cortex. This might be a bit too much detail for you, but it's worth having it anyway. It's just there, so yeah. And it's also expressed on interneurons or inhibitory neurons. So the pyramidal cells are described as the major excitatory cells in the brain, the glutamate cells, and in layer 5 that's a major sort of output layer of the cortex, but they're also found on inhibitory neurons. So it's worth telling you this because something's accounting for these decreases that we've seen with the fMRI. So is it a direct effect of psilocin stimulating 2A receptors on the interneurons, the inhibitory interneurons, which then shut off the excitatory pyramidals, or is it an indirect effect where you excite the pyramidals and they project to inhibitory regions which have a shutting off effect there, for instance, around the thalamus or in the basal ganglia? A bit too much detail, but let's get back to the brain network stuff. So what about the effects of psilocybin on functional connectivity? So I've already talked a bit about functional connectivity, and I've described how really this measure is about synchrony or coherent activity, coupled activity in different regions of the brain. So different regions, their activity fluctuating in synchrony, and how that synchronous relationship may change under certain conditions, like with the injection of a psychedelic drug. So what happens to functional connectivity using the medial prefrontal cortex as our region of interest and looking at where in the brain activity is synchronous with the medial prefrontal cortex, first getting that map, which is shown here, and we see the default mode network because the medial prefrontal cortex is a major node of the default mode network. So you can see the medial prefrontal cortex is in red, the orange is the regions in the brain where activity is synchronous with the red, with the medial prefrontal cortex. So then what happens when you give psilocybin? Well, you see a major hub of the default mode network decrease in connectivity, essentially become decoupled from the red medial prefrontal cortex there. So that's highly unusual to have a region which is usually tightly coupled in activity, tightly synchronous with the medial prefrontal cortex working together. You see the top images as the same system. Inject psilocybin, you get this very profound decoupling of this network. So that was when we used the medial prefrontal cortex as our region of interest. When we looked at the hippocampus, bilateral hippocampus. Here are the regions where activity is synchronous with the yellow there. I should credit Luke Williams for this analysis because he helped me out with this. So you can see the network there where activity is synchronous with the yellow, which is our region of interest, the bilateral hippocampi. And then when you inject psilocybin, you see this is really quite a dramatic finding because every node of the default mode network, massive effect in the posterior cingulate cortex. Let me see if I can get my pointer back. Well, I think you know where the posterior cingulate cortex is now. I hope so anyway. I pointed at it 3 times. The lateral parietal regions and the medial prefrontal cortex. So all these regions which usually have synchronous activity with the yellow there, they're usually part of the same system, all become decoupled, all become desynchronous, significantly desynchronous with the yellow, this major subcortical node in the default mode network. So what does this mean? Why is this network associated with the sense of self kind of becoming sort of disintegrated? Surely there's a story here that we could offer to explain these effects. Well, before I give that, this result shows a relationship between connectivity between the hippocampus and these default mode network regions. Posterior cingulate cortex here, that's the posterior cingulate cortex, medial prefrontal cortex. So the stronger the connectivity between the hippocampus and these default mode regions, the more frequent we either think about our past or think about the future. So there's a strong relationship between connectivity here and temporally extended or dexterous thinking, the ability to look far back or look into the future. So obviously there's something about this relationship, this network, which explains to some extent time perception, or the ability that we have to think about the past and the future and yet have an anchorage in the present. So, are these results evidence of some kind of ego disintegration? So we're seeing decreases in activity in the default mode network, this network associated with the sense of self. We're also seeing decreased connectivity within the network, or a decoupling within the network. And we have people saying things like, that was real ego death stuff, a total dissolving of the ego boundaries only existed as a concept, as an idea. I mean, that's quite a profound statement, you know. So you would assume that something quite profound is going on in the brain. Perhaps it's this that's accounting for those very profound effects. So interestingly, meditation, mindfulness meditation, decreases activity in similar regions of the brain. You can see they're similar with those increased with psilocybin. Also, a number of effective treatments for depression, suppress medial prefrontal activity. You see the medial prefrontal cortex circled in a lot of those images. So SSRIs, first-line medication for depression, decreases activity in the medial prefrontal cortex. CBT, cognitive behavioural therapy, so psychotherapy, decreases activity there. Sleep deprivation, effective treatment for depression, decreases activity there. ECT, a very effective treatment for depression, decreases activity in the medial prefrontal cortex. Placebo, when that's effective, it decreases activity in the medial prefrontal cortex. Deep brain stimulation, or the actual targeting of regions in the brain, applying electrical activity to shut off activity in those regions, that decreases medial prefrontal activity. That's the target site of deep brain stimulation. Also ketamine, which is emerging as an effective treatment for depression, at least in the short term, that decreases medial prefrontal cortical activity. So just to give you a nice good news story, one of our volunteers, she wasn't depressed, but she reported, as a number of ours have, positive mood effects after her experience. So she said, after her scan in Cardiff, that ever since Thursday, I'd say I found it much easier to engage in the moment, to attend to the here and now, whether this be watching water in a fountain or sitting in science talks and meetings this morning. There were some fountains in Cardiff and the water was being blown by the wind, allowing the sun to highlight the spray. I could have watched it for ages, somehow the beauty was enhanced. Whatever it was, it's lasted like the sun shining through the leaves this morning. It made me slow down my walk to work and enjoy the experience of it flickering over my face. So we had quite a few of these positive mood effects after our volunteers' experiences with psilocybin, and it's often described as an afterglow that they report. On the basis of this, and our imaging results and the overlap with the depression data, we we made the hypothesis that psilocybin may be effective in depression, and we wrote a grant proposing this as a research study, which fortunately we've been successful in. So this is a study that we will begin probably early 2013, giving intravenous psilocybin for depression. So it's going to be quite a major undertaking, but potentially something worthwhile. So, decrease between network competition with psilocybin. This is a bit high level, I'm not sure if I really want to go into it too much. It's essentially tying in with the disturbed ego boundaries reports. Essentially, I've already described this competitive relationship between activity in the default mode network and that in the task positive or attention network. And that explains, as I described, how we can only ever really daydream or focus our attention. And what do you see, for instance, this image on the right there shows that in people undergoing nicotine withdrawal, they're either given placebo or nicotine replacement therapy. And when they're given nicotine replacement therapy, you see a sharpening of this competition between the two networks. And they also describe being able to focus better. If you give them placebo, they say they're a bit muddled, and there's actually a strong relationship between the sharpening of the competition between the networks and their cognitive capacity or ability. So the stronger the competition, the better they're able to focus, the better their cognition. Where do you see, in what cases, in what scenarios do you see decreased competition? Well, you see it in schizophrenia. A number of studies have shown that. Interestingly, you see it in early psychosis, or patients showing signs of being at risk of psychosis. You see this decrease in competition between the networks. You actually see it in deep meditation as well. It's worth considering that in some state of deep meditation, you won't be, some people may describe it, you won't either be daydreaming or focusing, you'll be in some kind of other realm, a kind of state of non-dual awareness, or this mode of meditation. This study here, actually, the experienced meditators were doing a certain kind of meditation called non-dual awareness, where they're supposed to kind of relinquish any subject-object differentiation, and they found decreased default mode network tusk-positive competition in those people in this study. So the hypothesis is that default mode network, tusk-positive network competition underlies this subject-object differentiation or ego boundaries. So you can see that the prediction would be that we would see decreased competition between these networks under psilocybin, which would potentially explain decreased subject-object differentiation or disturbed ego boundaries. And I could cut a long story short by saying that's what we found. So here you can see in blue the tusk-positive networks, in red the default mode network. In blue the bar in these charts show increased connectivity, which is the same as saying decreased competition between the two networks. So that was highly significant under psilocybin. There was also a relationship between the subjective effects of the drug and this kind of blurring or melding together of the two networks. And you can see it more clearly here with the time course on the left. This is under placebo. You can see that the activity is orthogonal or unrelated. After injection of psilocybin, you can see it's as if the two networks are behaving together. They're kind of blurred together, they're smeared together. And you lose that differentiation, you lose that competition, so you lose that thing that we have during normal waking consciousness, where we either daydream or we engage. So summary hypotheses: default mode network activity and connectivity is related to constrained introspection and agency, or if you want another descriptor, another way of describing it, it will be the narrative self. And that ties in with things like people saying, that was real ego death stuff, total dissolving of the ego boundaries, I only existed as a concept, as an idea. Default mode network versus tusk-positive network competition is related to subject-object differentiation or ego boundaries, and when this is disturbed, there's a disturbance in ego boundaries. So here you could look at psychedelics as a model of psychosis, early psychosis, I should say. I should put a strong emphasis on it being a symptom seen in early psychosis, so really pre-delusional psychosis. And arguably, although this is quite a challenging thing to say, maybe in certain states pre-delusional, which are potentially even non-pathological. You know, something's changing, the world's becoming strange, but these people don't have a fixed delusion. They might just be experiencing the world as strange. And similar to some extent as people might experience on psychedelics and even in certain deep meditations. And so what is this disturbed ego boundaries thing all about? Well, it ties in with these reports of things like, it was certainly quite difficult at times to know where I ended and where I melted into everything around me. So this is from one of our volunteers on psilocybin. So very, very briefly, I'll talk about the ongoing MDMA fMRI work we're doing. So really I've just got 3 slides on this, and they're very brief. So this is a study in 12 healthy volunteers. We'll scan males and females. All will have previous experience with MDMA. It's a double-blind, placebo-controlled design. There are 2 scans, they're 7 days apart, and the order is randomized. So what I'll talk about very briefly, the only analysis that I have of 9 completers so far in the study refers to this autobiographical memory recollection task that we do in the scanner. So what happens with that is people provide us with 12 personal memories. We ask for 6 of their very worst memories. If they have traumatic memories, then include those, just essentially their worst memories. 6 of those, and 6 of their very best. 6 of the worst might be things like remember the crash on the Westway, something like that. We sometimes ask that our volunteers encrypt them so that they're meaningful for them but not for us. We don't want them being self-conscious. We also want them to give us truly their worst memories. We don't want them to hold back. So there may be things that they might be embarrassed, ashamed about, that they wouldn't offer us. So if we say, well, encrypt them, then they're more likely to give us truly their worst memories. And we ask for 6 of the very best memories. Things like remember the beach and Saturday. Okay, so here's our analysis coming up. This is looking at where activity to negative memories. So it's activations to negative memories versus baseline are greater under placebo than MDMA. So there's relatively more activation under placebo to negative memories in these regions than there is in MDMA. So equally you could think of this as reduced activity to negative memories under MDMA. What's interesting is that we have bilateral insular regions where activity is less under MDMA. These are related to interoception or internal sensation, pain especially, even psychological pain. And also this subgenual cingulate cortex region is strongly linked to depression. It's actually the target of deep brain stimulation for depression. And actually if you induce low mood, you see an increase in activity here. So what we're seeing with MDMA is a reduction in activity in this region linked to low mood and depression. So that's interesting. It fits what we know about this region and its function. Also, when we looked at activations to positive memories versus baseline, we saw greater activations under MDMA than placebo, so a reverse pattern. So there was subjectively a positive bias coming from our volunteers. This is what they were reporting, that their negative memories felt less weighty somehow, and less, they sort of cut less. And actually they could sort of think about them in a more philosophical way sometimes. And the positive memories felt especially nice. They felt even more positive somehow. So there seemed to be quite a strong, at least subjectively, positive shift in positive shift towards a positive bias in their memory recollections. And just to give you a final sense of how that was described with one of our volunteers, he said, an interesting thing occurred to me in the MRI. When I reached back for the bad memories, they did not seem bad. In fact, they seemed to have been fatalistic necessities for the occurrence of the later good events I had to recall. The whole nexus of events became interwoven, a part of destiny that allows for the good to happen only because of the bad, making everything good and worthwhile. That's quite a happy tone to finish on. So that was shortly after the experience. And certainly strong shift in appraisal of things. So finally, some acknowledgements. The silverback, my mentor and a very important, crucial character for me. I wouldn't be here if it wasn't for him. Also Amanda Feilding, who's been crucial in setting up this research program that we're doing, a very important initiating partner in that work. And the Beckley Foundation that provided a lot of support for what we're doing, most of the support. Also the Hefter Research Institute in the States, MAPS, the Multidisciplinary Association for Psychedelic Studies, and the Neuropsychoanalysis Association. Lots of people to thank, but thank you very much for your attention.
Thank you very much, Robin. That was an extremely fascinating talk and brilliant research. Long may it continue. We have some time now, about half an hour, for some Q&A. We'll take some questions from the floor. I'll just take the first prerogative, my kind of prerogative as the host, and ask you a question. I was wondering what you might think about some of the kind of classic scholars and their notions. I mean, obviously quite crudely, about kind of brain function, and particularly in light of the mystical experience. So people like Bergson and Huxley, and their notions of the brain as a filter, and how if you kind of reduce the action of the brain, you have a kind of a more of an expanded experience. So take the classic Blakean quote, if we were to, if the doors of perception were cleansed, now we'd experience everything as it truly is, infinite.
Yeah.
Obviously that's much more of a kind of poetic summation of this, but it does seem to tie in with some of the neuroscience that's coming through now.
But it does seem to tie in. One of the another ways of thinking of this, I know I've gone over it quite a lot, with this default mode network and its function, that we see decreases particularly in this network, is that it suggests that the high level of activity in this network, its function is to keep our experience of the world very steady, very stable, very familiar. So if you lose that function, then things become unstable and chaotic. And so essentially you could think of, even though it's high activity in those regions, as that functioning in a way as a filter, it's a kind of constraint on the dynamics of the system, keeping it within certain bounds so that you experience familiarity really in the world. So I think in terms of where's that filter, you might think of those regions in that network.
Thank you, Ron. We'll open up to the floor for some questions.
Thank you at all. I'm really interested in developmental neuroscience, and I was wondering if you thought that if you took lots of psychedelics when you were very young and your brain was still forming, do you think that you could sort of permanently disrupt the level of connectivity in your default mode networks, and then when you grew up you'd be less likely to be depressed and neurotic? So it would be a good idea.
It's a hypothesis, I don't know if you can ever test it.
But do you think that is?
I think there's probably something about the system organising itself which maps onto maturity, and there's actually quite good evidence that the default mode network does increase in connectivity and the nature of its connectivity changes, and that maps on with maturity. So I think it's potentially quite dangerous if you're giving psychedelics to someone who's developing, that they might not mature. It's just another way of looking at it. The system might not organise as it usually does, but equally a system can become ossified in its level of organisation. It can become too rigid. So when you think about psychedelics being potentially useful for creativity, which is another hypothesis, then that might be how it may destable a kind of overly organised system.
But it could go sort of too far in the other
Well, it
direction.
could if you could induce an instability that may, for instance, increase, it could precipitate some psychiatric disorder potentially, if it's not done in the right way with the right support and such like. So, yeah.
And then Julia.
Yeah, I'd actually like to turn that question round, and one of the various things you were comparing to this state of lacking the anti-correlation was psychosis and other sort of pathological conditions, but actually very early childhood is another situation where networks aren't really that developed, and so perhaps it's not so much more the case of thinking of what would happen if you took psychedelics as a child, but that being a child is a lot like being on psychedelics.
Absolutely. I think that's really true. And in terms of the connectivity pattern, it's relatively less. Connectivity in the default mode network is relatively less if you go back. People often just describe the experience as being regressive. It could be regressive in a kind of, let's get my terms right, ontogenetic way. So in terms of development within your own lifetime. It could also be regressive in a phylogenetic way. So it could be like our style of consciousness is becoming how it used to be as we were developing as humans, as a species. So it's definitely, I think that's true.
Yeah, hi Robin. Connected to that slightly, I'm wondering, because as it's been mentioned before, there are these similarities with, as the gentleman said, pathological states, how far along you are in differentiating between what would be seen as a destructive, generally accepted state. What is it that is different scientifically? What is different going on between something that's destructive and the higher states of meditation? And the other part of the question was, most of this, well, seemingly the experiments are the person, the individual is in a kind of passive state. Are there similar brain scans available for monitoring peak experience? Because there's often the accounts of peak experience in athletics, for example, is of a certain dissolving between right and wrong and this kind of other mind taking over. So is there a way of doing the same kind of brain imaging?
It's about capturing that peak experience. So it's whether you can have them in the scanner having the peak experience. That's
They could be in
that's the
state.
limitation.
Yeah.
You'd have to get them in quickly.
There could be this maybe beforehand and after, in like for example a 2 or 3 year period, an athlete will seem to continually have this peak.
Yeah. If it's I mean I think it would have to be state. You'd have to capture the specific state.
Before and after.
Yeah.
But as far as schizophrenia.
But the first was a really tricky question to answer, and I don't know, because people often describe the experience as being paradoxical, that it can be both euphoric and hellish at the same time almost. As I imagine that the brain effects are the same, but then what does that mean? And I think maybe the dynamics change so that you can have this paradoxicality that can be one moment hellish and one moment heavenly, and maybe even at the same time. But perhaps in moments of sheer terror and horror, there may be a certain activation profile to that. And also, the limitation of these blobs on brains is that you're just getting blobs on brains. You're not seeing any dynamics there. And so if we were to look at the reality, we might see, phasic activity happening during a particular moment of terror. Maybe there's some amygdala activation during that. Maybe there's something about the breakdown in this high-level network and its maybe tonicity, or the fact that it's always there, highly active, that allows phasic stuff to happen, that we can be shocked by this and that.
Is there any change between someone who's had a fearful approach to it, totally terrified, and someone
As
who's...
far as we can tell, no. So when we did those correlations, looking at valence, we didn't find anything.
I think you've already answered some of the questions I was going to ask. It was really about the context of the experiment being in this big machine, and just wondering whether there are other possibilities of there being mobile scanners, such as built into a helmet or something like that, which you could wear for a longer period of time, and perhaps plug into your own laptop and give results. Maybe a larger number of people wearing these kind of portable scanners and feeding, doing a kind of bigger, more extensive experiment, and also giving results in different contexts. I think being in a big machine sort of it's not the normal context in which people, I want to do one, all kinds of states, in partying or whatever, wearing some kind of, maybe you're aware of, is it something that is portable, that gives some aspects of the results you're getting, or do you need to have some very expensive equipment?
Even EEG, the brainwave measures that you may be familiar with, they're sensitive to motion artifacts. Virtually every imaging modality is sensitive to motion artifacts, so it's very, very difficult. The technology's not there yet. It sounds like a wild idea, but
if in years into come it would be possible to...
If it was possible, it would be brilliant.
You would embrace that.
Oh, absolutely, yeah. It's always changes it's often changes in technology which lead the insights.
Is it the old alpha wave machines?
Yeah, that's the EG, yeah.
So I've got a question. Thanks for your talk, Robin. But before the question, I'm just going to say quickly about the question about children taking tryptamines.
Okay.
There's been quite a lot of research in Brazil about people in the ayahuasca churches, in the ayahuasca circles there. And first they legalised it for people over 18 after a whole battery of tests, and then they extended that to pregnant women and children. So the scientists there, and it was lots of tests, it wasn't the same things that Robin was doing, but they found it was alright, and some of the coolest people I know have been drinking ayahuasca since they were in the woods.
That's proof.
But the question I was going to ask was about nicotine, in fact. It went completely over my head, that diagram.
Yeah.
But looking at what they do in shamanic setups, where they're taking ayahuasca, which is very similar to psilocybin in its molecule, along with hape, along with snuff, along with nicotine. I was wondering how those two drugs would work together and what kind of things they bring out.
DMT's very similar molecularly, pharmacologically to psilocin. But ayahuasca is a more complicated thing because you've got the MAOIs in there. So simple answer, honest answer, I don't know, but there are those similarities.
No, but I'm talking, sorry, I think I made myself clear about the nicotine,
the nicotine.
And the effect of nicotine plus, say, psilocybin.
Right, okay. Well, it may suggest that, you'd think if you were to smoke, it may kind of settle the intensity of the psychedelic experience. I don't know subjectively if that's true. I'd have to ask someone who's that would be a first.
I can tell you about that, Robin. Actually, it was just with the Huichol Indians, and they wear cigarettes in their hats. So when the visions get too strong, you smoke a cigarette, and that helps control the visionary experience.
Yeah.
So subjectively, there's some element to support that.
Thanks, Robin. I was just thinking, say there were to be like psychedelic clinics or health farms or whatever. Talking about age again, what about the upper limit? So is it safe for the elderly if you were to say find a 70 or 80, 90, whatever age year old who wanted to take mushrooms. Do you think, is there any research into how safe that is physiologically or whatever system you're looking at? Is there any knowledge really about, you know, is there danger, or is it even easier? I don't know.
Well, I mean, we've monitored physiology when we've given intravenous psilocybin. Heart rate and blood pressure can increase quite significantly, quite markedly. But it doesn't seem to be the drug leading the change. It seems to be to do with a kind of anxiety, uncertainty of entering that state and it being so novel and strange. So equally, we've had volunteers whose heart rate hasn't increased at all, and sometimes actually decreased a bit. So it doesn't seem to be the drug leading the physiology. So it would depend really on the health of their cardiovascular system, for instance.
The shame was to miss out, really, isn't it?
Yeah, I guess so. Of course, people are giving psychedelics for end-stage cancer and anxiety around death. And physiologically, I think it's been well tolerated. I don't think there's any special risks other than the natural considerations and screening procedures that you go through before giving anyone a psychedelic.
Yeah, thanks, Robin. What was I going to say? I guess I was curious. Just one of the things I was curious about was that psychosis, sort of early psychosis and deep meditation had quite similar results. So that was just something I was just quite curious about, because obviously one of them was quite positive. Somebody mentioned that before, so I was still, because I work with schizophrenics, and I do find that they often can tap into lots of unconscious material that can be sort of verging on spiritual as well. And so I was interested in that, and also just long-term effects of. Have you ever done sort of looking back at the people that you trialled and seen sort of whether there was any changes in the brain, sort of a month on, 2 months on, a year on?
We haven't looked at any brain changes. I mean, there's some important work looking at long-term subjective changes and changes in personality. For instance, there seems to be significant increases in trait openness post psychedelics, and I mean that seems like a potentially quite positive thing, that openness is changing. That might map onto creativity and general well-being potentially, and there seems to be a correlation. But equally, openness can verge on things like schizotypy potentially. So, like all of these things, you have to appraise them soberly. So the other issue about, I mean, it's kind of relevant, the meditation and psychosis thing. Important thing to say is that I think where the phenomenology overlaps would be in early psychosis, and you might even say pre-psychosis or the at-risk mental state. And it's there that people might describe things like disturbance in their sense of self and experiencing the world as strange, labile emotions maybe. And also these mystical, spiritual type experiences. They're quite common in people experiencing a kind of strangeness of the world that happens sometimes at the onset of a psychosis. So I think that's where the mapping is. It's odd to think that you could have something which is potentially so devastating, psychosis, and something that people do often just to relax, meditation, and that there's a similarity in what's going on in the brain. What it may, to me, I mean, this is a challenging idea, but people are always emphasising the importance of mediating a psychedelic experience. But it's a challenging experience, you have to have a positive setting, and really you should go into it with a certain mindset, or at least you should be prepared for dealing with difficult material. If you were to say that to someone in the at-risk mental state and give that level of care and support to try and mediate them through them experiencing the world as strange, then that could be a positive way of dealing with that kind of naturally occurring psychedelic experience, if you want.
Go on, Chris, quick one.
You talk about, some of your subjects talk about cogs, and you talk about hubs and blobs and brains. Do you think we're heading towards a kind of mechanistic perception or conception of the brain? And is the mind limited to the brain? I mean, is this kind of research limiting the mind to the brain?
I would say it's doing the opposite, really. It's sort of expanding our knowledge base. I don't think there's anything to be worried about in terms of having a mechanistic account of nature. There's systems in nature which follow laws, and we're just trying to identify those laws, those rules, and to better understand nature. I don't think we lose any of the magic. I don't know if maybe I'm misunderstanding the question. I know there's certain anxiety sometimes about finding stuff out about the mind through finding stuff out about the brain, but I don't think there's anything to worry about in losing some of the magic of the mind through understanding its basis in the brain.
The machine seems a bit, I only think the machine seems a bit limited in that it doesn't grow maybe in the same way that nature grows.
It's a mechanical explanation, I guess. There's systems in nature, and
we talk about spiritual experiences, and to kind of mechanise those spiritual experiences, it doesn't quite add up from my personal experience.
It doesn't know what the thoughts are, the thoughts themselves are.
Well, you can't kind of have a formula for creativity.
I guess another maybe analogy is you could have a mechanistic model of the cosmos, or you could have a very loose one. And I think the mechanistic models that we have a closer to our learning, through our science.
So do you think we're going to stick with the mechanistic?
Unfortunately, yeah, I think,
Do you think we could
get
a
twist?
I think,
the map isn't the territory.
In order to understand things, we offer a model that explains it, and the models are often mechanistic, and they seem a bit crude. And that's where we are. It seems a bit crude. I think that's what it is.
Just, I know that people want to have a glass of wine and all the rest of it, and Owen wants to watch the football, so just two last questions.
You showed the graph showing the competitive relationship between the task positive and defaults.
Yeah.
And with the nicotine, it showed that the withdrawn state was sort of a chaotic relationship between the two, whereas the psilocybin showed actually a complete inversion. It was very close correlation. I was wondering, with the other states that you said were similar, the meditation and pre-psychosis, what was the relationship there on those graphs? Because that seems quite significant to me.
The measures would have to be, I guess you'd have to have them in the same volunteers, in the same setting, in order to make a genuine comparison. That's the problem. It's not really an absolute measure, so you don't really get a quantitative figure where I could say the effect was as great in these different states. So that's why I can't really answer that, unfortunately.
So that does seem something to investigate further. If you give someone psilocybin and get them to meditate and get them to have psychosis.
Yeah, and actually, get them to have psychosis. Throw that one in as well.
Final question then.
Yeah, hi. Your most interesting discoveries of reduced activity in the brain, would you expect to see that in a dying brain, for example, and might that explain near-death experience?
Yes.
Just one other thing as well. You looked at placebo as well. Did you find any individuals that were, with placebos, being able to mimic some of the effects?
Both really good questions. I mean, the first one, yes, that's a strong hypothesis that actually the near-death experience phenomenologically does share some quite strong similarities to the psychedelic experience. And you would offer the hypothesis that these regions which receive so much metabolism and blood flow normally, that those regions are going to experience a decrease, when you're dying. I think is a very strong hypothesis. So this may be modelling what happens to the brain, what happens to the mind when you die. I think it may well. Now the other one, that's such a deep issue that I lost track. The other
was the placebo.
The placebo, yes.
Individuals that reacted.
Yeah, well actually I ran a little analysis on the placebo responders. Placebo responders versus the non-responders, and we saw some decreases, and they were in quite similar regions. So that was quite neat. I thought about writing a paper about it. there's just so much to do. But
Because we have DMT naturally in the brain, so,
yeah, maybe there was some endogenous DMT release that's accounted for that.
Yeah.
On that note of near-death experiences, there's some beautiful Tibetan artwork downstairs looking at the bardos and the journey through the near-death or death experience, perhaps even the psychedelic experience. Thank you everybody for a very stimulating evening. Please do come again and give Robin a great thank you.